Hello everyone, my name is Meghna and today I and my group partner Gurpreet are presenting a comparative analysis of green tea and black tea extracts versus antibiotics in terms of their antibacterial efficiency. This research specifically examines their effects on E. coli and S. aureus bacteria, which are two common pathogenic bacteria. This project was completed under the guidance of Professor Nurul Hassan. Antibiotics have been critical in treating bacterial infections, but with the growing issue of antibiotic resistance, alternative solutions are urgently needed. Pathogens like E. coli and S. aureus are becoming harder to control. Green tea, known for its rich catechin content, has shown promising antibacterial properties in previous studies. Our research compares these properties to traditional antibiotics such as ampicillin and erythromycin to see if the tea extracts could potentially replace antibiotics as a sustainable and natural alternative. Talking about the purpose of the research, our study had two main objectives. First, to compare the antibacterial efficiency of green tea and black tea extracts to antibiotics when tested against E. coli and S. aureus bacteria. Second, to investigate if tea extracts could serve as a natural alternative to combat antibiotic resistance. We hypothesized that tea extracts might inhibit bacterial growth as effectively as or even better than conventional antibiotics. This research could have significant implications for finding sustainable solution to the antibiotic resistance crisis. Next is the methodology. To start with, our methodology involved preparing different media to culture bacteria. For McConkey agar, we dissolved 12.5 grams of powder in 250 milliliters of water, boiled the mixture, and then sterilized it using an autoclave. Similarly, mannitol salt agar was prepared by dissolving 29.18 grams of powder in 250 milliliters of distilled water by following the same steps. We also prepared a phosphate buffer solution by diluting 0.625 milliliters of stock solution into 500 milliliters of distilled water, which was dispensed into test tubes for later use. These preparations ensured a sterile environment for our experiment. Next is the preparation of tea extracts. For that, we prepared three concentrations of green and black tea extracts: 1%, 5%, and 10%. For the 1% extract, 1 gram of tea leaves were steeped in 100 milliliters of boiled water for 5 to 10 minutes. The process was similar for the 5% and 10% extracts, but with 5 grams and 10 grams of tea leaves, respectively. We followed specific temperature guidelines: 70 to 80 degrees Celsius for green tea and 90 to 100 degrees Celsius for the black tea to ensure optimal extraction of bioactive compounds. Now our media was prepared earlier. So next is media plating and bacterial streaking. We used sterile agar plates for the media plating. After adding 100 microliters of bacterial sample to the agar surface, we spread it evenly using a sterile spreader and incubated the plates at 37 degrees Celsius for 24 to 48 hours. For bacterial streaking, we used sterilized loops to streak bacteria across the plates using quadrant and radial methods. This allowed us to isolate colonies and ensure proper dilution for accurate observation. Next is the dilution. So, for the dilution, we started with a 1:10 dilution by transferring 1 milliliters of bacterial culture into 9 milliliters of phosphate buffered saline. From there, we performed serial dilutions, labeling tubes from 10 raised to power 1 to 10 raised to power 8. Each dilution step involved transferring 1 milliliters from the previous dilution into 9 milliliters of buffer. This process helped us achieve precise bacterial concentration for testing the antibacterial effects of tea extracts. Our antibacterial testing involved pouring sterilized agar into petri dishes, spreading diluted bacteria, and adding tea extract solution to the plates using wells or discs. After sealing the layer with an optional thin agar overlay, we incubated the plates at 37 degrees Celsius for 24 to 48 hours. Finally, we observed inhibition zones and counted CFUs to assess the antibacterial activity of tea extracts compared to the antibiotics. Next are the results and our observation, which will be discussed by Gurpreet Kaur Bhangu.
Hello everyone. As my friend Meghna discussed about the previous slides, now I am going to continue with the observation and data tables. So here is the results of our study. So firstly, we chose different bacteria like E. coli, Ascaris, and we chose different dilutions like 10 raised to power 5, 6, 7, and 8. In the tea extract, it was 1%, 5%, and 10% to check the antibacterial efficacy against the antibiotics. So we chose two different green tea and black tea. So in the last two, here are the CFU count of our results. So as we know that green tea is better has antibacterial activity as compared to black tea. So with the 10% green tea showing the most significant effect against E. coli and Ascaris. Uh, Moreover, for black tea extract, antibacterial activity was less pronounced, even at higher concentration. So results indicate that the green tea has more active compounds like catechins, which contribute to its stronger antibacterial properties. So, moreover, additionally, we can say that antibiotics like ampicillin and arthromycin exhibited the most significance zone inhibition zones that I am going to discuss in the next slides. So here is the continue table of the observation and data results for the tea extract against bacteria. So, so now we are going to discuss about the black tea versus green tea. So according to the final observations, we can say that antibacterial activity has to highest antibacterial activity against bacterias. So 10% green tea shows the highest activity and 5% green tea shows less and 10% black tea shows uh, more less than the 10%. So according to the key findings, 10% green tea extract demonstrated the highest antibacterial activity for both E. coli and Ascaris. So observations from dilution numbers like from 10 raised to power 1 to 10 raised to power 3 2 for E. coli, maximum inhibition zone observed with 10% green tea. So in the previous tables, it was shown like in the serial number 33, 37, 41, and 45. So for SREs, a similar trend with 10% green tea showing the best antibacterial efficacy. So according to the conclusion, increasing concentration enhances antibacterial properties. Now continue with the effects of antibiotics on E. coli and S. aureus. So here is the procedure for that. Firstly, we prepared media plates like McConkey agar and MSC. McConkey agar sports E. coli, which is gram negative and produce pink colonies. So in terms of MSA agar, it sports S. aureus growth, which is gram positive, producing yellow colonies. Then we inoculate plates, evenly spread E. coli on McConkey agar and Azores on MSA agar using sterile techniques. Then we applied the antibiotics and then again incubated the plates at 37 degrees Celsius for 24 to 48 hours. After that, we measured the inhibition zone, in which we recorded the diameter of the clear zones around antibiotic discs to assess bacterial susceptibility. So for this, we calculated bacteria reduction. We compared the CFU counts or inhibition zone to determine the antibiotic effectiveness. Here are the results for the ampicillin antibiotic, which we use against E. coli, and arthromycin antibiotic for S. aureus. So now let's move to these results. Starting with ampicillin, which was tested against E. coli, the zone inhibition ranged from zero point 0.6 to 1 centimeter with an average diameter of zero point 0.8 millimeter. This confirms that ampicillin has moderate effectiveness against E. coli, reflection its actions against gram-negative bacteria. Moving on to arthromycin tested against S. aureus, the inhibition zone were larger, ranging from 1.5 centimeter to 1.7 centimeter, with an average of 1.6 centimeter. These results highlights that arthromycin's superior effectiveness, particularly against SREs as a gram-positive bacterium. So inhibition. So now continue with the inhibition zone test, green tea and black tea. So here is the procedure for that. Firstly, we prepared media plates, like we poured McConkey agar into 6 plates and MSA agar into different 6 plates, and then we allowed to solidify it. Inoculate the test organism is the next step. So firstly, swab E. coli and S. aureus cultures evenly across the respective agar plates to form uniform bacterial lawn. Apply tea extracts, punch wells into the agar using cork burr. Then pipette 20 to 30 microliter of 1%, 5%, and 10% green teas extract into each well. Next step is to incubate plates. Incubate all plates with 37 degrees Celsius for 18 to 24 hours. Observe results, measure the inhibition zone, and record the results for green tea and black tea extracts. So here we continue with the data and the results that we previously discussed in the slide. So here is the bacteria we use, E. coli and S. aureus, concentration 10%, 1%, and 5% as usual. Expected zones of inhibition was 1.5 to 2 centimeter for E. coli, in which we use 10% concentration, zero point 0.8 to 2 centimeter for S. aureus for 1% concentration, and continue with these. So if we talk about the E. coli for 1% concentration, the expected zone of inhibition is zero point 0.3 to 0 0.6 centimeter. So here are the results which we have done for the inhibition zone. So here are the final observation. 20% green tea extract shows the highest antibacterial activity. Uh, in order, like 20% green tea, then 10% green tea shows less, 5% green tea, 10% black tea, 1% green tea, and then 1% black tea. So if we compare the results with the green tea, black tea, and antibiotics, antibacterial efficacy, antibiotics, ampicillin and erythromycins show significantly larger inhibition zone than tea extract. Then, if we talk about the tea extracts, green tea more effective than black tea due to higher levels of active antibacterial compounds. Bacterial target tea extract work together better against Ascaris gram positive compared with E. coli. So here are the all comparative results for green tea, black tea, and antibiotics. Last we compared in which we noticed that green tea at 10% approaches arthromycin in efficacy, but it is still less effective. Black tea at 10% produces a smaller inhibition zone compared to both antibiotic. So in this slide, we discussed about the conclusion. Green tea extract demonstrated strong antibacterial activity against E. coli and Ascaris with 10% concentration, showing the highest effectiveness. Green tea was more effective than black tea, particularly against Ascaris, while tea extract were less effective than ampicillin and erythromycin. 10% green tea came close to erythromycin and antibacterial efficacy. These findings suggest that green tea could be potential natural alternative for bacterial control. So these are the references that we used for our research purpose, and thank you for watching our presentation. Hopefully you will like. Thank you.